So it will be a set of 3 lectures whose basic theme is sensing via chemical signals. That's what chemosensing means. And chemotaxis is the response that cells or individuals have to specific chemical signals. Please feel free to interrupt me at any time if you feel that's something that doesn't sound quite understandable. So I'm really willing to give you all the details and to understand what the problem is. So our basic starting point is about sensing in general. So sensing is actually one of the most important features of living organisms. Senses are our interfaces to the outer world, and represent also our the way in which we humans communicate with each other. So the importance of senses actually becomes very, very clear when we think at what happens when we lose them. So as you might know, there have been recently several experiences in sensorial deprivation. So for a limited amount of time, reducing stimuli, external stimuli, is something which makes you feel good. Currently, there are places, just recreational places, just like spas, which offer you the experience of staying an hour in an environment which is you float actually in a tank, and there's no noise, there's no light, and this is quite rewarding and makes you feel better, lowers your anxiety, and it's all good. A certain degree of sensory deprivation helps you feel better. But then, as you might know, if this experience is prolonged, if you undergo sensory deprivation for 24 hours or more, then it starts to be a real bad experience. Actually, sensory deprivation has been used over the years as a means of torture for prisoners. Only visual senses deprivation, that is, if you stay in the dark, after 24 hours you start experiencing hallucinations. And these hallucinations become more and more severe, and then they can even lead to mental troubles after a while. So from the clinical viewpoint, this thing is very well known. So there are, for instance, special pathologies that are explicitly related to the fact that senses are deprived. One of them is the Charles-Bonnet syndrome, which is actually a syndrome that affects many of the elderlies, and basically 30% of the people who have severe sight impairment. As time goes by, they start experiencing hallucinations. They know that these are hallucinations, so it's not a mental disease. It's just a problem that the eye, in absence of stimuli, starts to see things that weren't there, because our brain, our senses are made for experiencing. And when stimuli lack, then the brain starts to rearrange itself in such a way that produces these false stimuli. This is true also for other sort of stimuli. One example is the so-called Ganzfeld effect from a German word, which means the whole field. If you are subject to a visual stimulus which is not in the dark, but you see a stimulus which is not patterned, say just like a continuous reddish background, maybe flickering. If you see that, after a few minutes you start having strange sensorial experiences, hallucinations, and you see things moving which are not there. So even a stimulus, if it's not patterned enough, leads you to a wrong understanding of what the environment is around you. And there are also auditory correlates of these effects. One is the so-called ear musical syndrome. There's people suffering from the syndrome, typically people who are deaf or have a hearing impairment. After this impairment starts, they hear music playing. Actually, one famous example is Dmitri Shostakovich, which was known to have this syndrome. So he was hearing continuous music, and then he said, okay, I'm taking this music and playing it down. That's what he said. So all this just is to motivate us that senses are very important to us. Without them, we would be lost. Of course, vision and hearing are sort of our favorite senses. We can easily make an idea of what happens if we lose sight, if we lose hearing. That's very common. But there's also one sense which is often referred to, which is the forgotten sense, which is olfaction. Olfaction is a sense that permeates our everyday experience. And we don't fully realize this unless we lose it. There is a pathology which is called anosmia, which means you cannot feel odors, which typically happens either on a genetical basis, but it's very rare, or after cranial trauma. And patients who suffer from this traumatic anosmia refer a whole range of problems that shed light on the importance of this other sense. For instance, they report that they lose their attachment to their husbands or wives, to their children. They don't feel anymore their children as theirs. And the origin is that we have this sense which is actually at the very unconscious level in ourselves. But it's very, very important. So this was a sort of introduction to let you appreciate how pervasive the importance of senses is in ourselves as humans. But in the course of the lecture, we will try to have a grasp on what senses mean at all sizes and scales in the living world. So let's start from very few basic questions. The first one is, how many senses are there? 5. Very good. 5 senses. Do you know who was the first one who said that there are 5 senses? It dates long ago. It was actually Aristotle who first made this classification. So he said there are 5 senses, which you know. So this classification is essentially based on an anthropomorphic idea of sensing, of course. There is sight, we have the eyes. Hearing, we have the ears, nose, mouth, hands. Okay? So this is basically a classification based on the fact that we have sensory organs, and these are very prominent in ourselves as well as in many mammals, which you can see, oh, this lion has a nose, the lion has ears. And then you go and then you think, the fish has a, fish what? Does the fish have a nose? Well, they can sense odors, right? Even if they are in water, which is pretty much different than in air. And they can feel many other things. But then you see, you start to thinking about different classifications. So maybe we should classify senses not depending on the organ, but depending on the stimulus. So let's try to make a physical categorization of stimuli and understand what the senses are. And you see that the things start to mix up a little bit. Okay? For instance, are touch and hearing different from a physical point of view? Well, you would say yes. You know? Yeah, sound is vibration of a medium. It's a mechanical phenomenon. And in fact, you can put sound, you can put hearing and touch pretty much on the same level. These are 2 instances of mechanosensing, sensing mechanical stimuli. You can sense the rigidity, the softness, compliance, the vibration in the air. It's just the medium which is different, the solid or aqueous or gaseous, but the stimulus is the same. So you can classify senses according to stimuli. And then you say, okay, there's mechanosensing, there's touch, and there's hearing, and there's kinesthesia. What is that? Well, Aristotle wasn't mentioning that. Is it a sense? You know what it is? Oh, it's very important. If we lost kinesthesia at the moment, we would be lost totally. Kinesthesia is the sense, the perception of your body in space. I mean, you know where your hand is even if you don't look at that. You don't have to look at your hand for everything. If you close your eyes and say, okay, touch your nose, you do it. Kinesthesia is the sense of our body. It also goes under the big name of proprioception, sensing yourself. And it's very important. And it's also something that is present at all levels in biology. So these three senses, you can put them all together under a single label, sensing mechanical stimuli, because kinesthesia is you sense the tensing of your muscles in your body, if they're tense or they're relaxed. This gives you information on where your limbs are, what your position is with respect to gravity, etc. Gravitation forces. Then there is a second class, large class, which is chemosensitivity. And this couples gustation and olfaction together, because gustation is chemosensing. Gustation on your tongue, well, there are receptors on your tongue that sense chemicals. And olfaction is sensing chemicals through your nasal mucosas. Actually, gustation as a sensation, as we feel it, is for a large part olfaction. Is retroolfaction. So it's sensing the smell, the odors of food you eat from inside the mouth through the pharynx. And then there is a third class, a large one, which is photosensitivity. Okay, we agree. Three stimuli: mechanical, chemical, and light. Okay? Unless you don't want to consider photons as chemicals, but that's quite a stretch. So you see, according to this classification, which is based on stimuli, there are basically three senses: the sensing of mechanical stimuli, etc. And then if you reason along this way, there are actually other senses, because there are other stimuli that you can feel. You can feel temperature, which is neither chemical nor light by itself. You can feel pain. And pain is something different from all this stimuli. And then there are animals which can perceive magnetic fields, birds, and others that sense very well electric currents, especially in water, as you may imagine. So you see, the senses are neither 5, nor 3, nor 7. There are many of them, depending on the way you just classify them. For our lectures, we will mainly focusing on chemosensitivity and olfaction, which is sort of its large-scale counterpart. But now, this classification makes perfect sense, but there is still another one that you might think of. You might think as a biologist, as a molecular, cellular biologist, right? And so what is sensing at the cellular level, at the level of a single cell? Does it make sense to talk about a cell's nose or a cell's ears? Well, actually, it does make sense. That's basically our starting point. Only that you take a completely shift of perspective. Rather than thinking about sensory organs, you think about the equivalent of something that is able to make contact with the environment. So how does a cell make contact with the environment? What are the biochemical structures that put you in contact with the environment? Receptors. So you can classify molecularly senses by the classes of receptors that they put you in contact with the environment. And there are a few of them, actually. I'm oversimplifying, as any molecular biologist would know, but basic classes are these ones. G protein coupled receptors. This is an enormous class of receptors which is present basically in all cells from single-cell amoebas to ourselves, and includes a vast number of potential signals that come in. So I might take the opportunity to write down on the blackboard what these receptors are. Just the wiper. No, that's too cumbersome. I just need to wipe the blackboard. Thank you. May I erase this? So these G protein coupled receptors are actually what are called 7-pass transmembrane proteins. So you take the surface of a cell membrane. These objects are basically made like this: 1, 2, 3, 4, 5, 6, and 7. This is the inside of the cell, and this is the outside of the cell. Then there is some signal which comes from outside, chemical, typically, but it might be light, because rhodopsins, our receptors for light, which are in our retina, are G protein coupled receptors. So stimuli come. If it's a chemical, it binds this receptor. This receptor changes shape according to the stimulus. This change of shape, which is called allostery in general, is transduced to the inside of the cell and modifies the chemical properties of this receptor with respect to what is inside the cell. And what is inside the cell for this particular receptor it's called G proteins. And then they typically come in a pair. They bind, this protein binds the receptor from the inside, and when the stimulus arrives, it unbinds. This is a very simple mechanism of transduction of information from outside to inside. Stimulus arrives, and then something inside changes as a result. And pardon? Why is that happening? It's happening just because the binding changes the shape of this protein and exposes a different electrostatic landscape inside. If you're asking about the physics that's behind it, the chemistry, it changes the shape and it exposes different residues inside the protein, the shape of the inside part. Okay, just mechanically, you might think that this changes from this state to something which is actually, I'm exaggerating, like this. Okay? It's in a helix which winds back and forth, changes shape and exposes different residues, changes the chemical properties, the affinities, and induces changes in the proteins that are bound to this receptor from the inside. Okay? Everything is in chemistry, is just electrostatic forces, van der Waals forces, every kind of force that you have inside a liquid medium. So this conveys the message. And you see that these G protein coupled receptors actually mediate lots of stimuli: vision, olfaction. 3 of the 5 tastes, the bitter taste, the sweet taste, and the umami taste. There are 5 kinds of tastes. Okay? Bitter, sweet, and umami are mediated by G protein coupled receptors. G protein coupled receptors are also sensing hormones. So for inside signaling, from within our body, there are signals that are sent from one organ to another through hormones, and G protein receptors are these hormones. And also neurotransmitters in our brain. Communication between cells, between neuronal cells, takes place using G protein coupled receptors. And there are many, many other examples that come along, including olfaction. The second big class of receptors are ion channels. Ion channels are channels that open and close at the cell membrane, often depending on some ligand binding to them. So ligand-gated ion channels are channels that open if there's some chemical signal from outside. The signal arrives, the channel opens, lets something in, then it closes. Okay, it's just like a gate. And ion channels respond to several chemicals, and they are responsible for the 2 other of the 5 tastes that we have. Tastes too salty and too sour are based on ion channels. And mechanosensing at a very basic level, sensing osmotic pressure, is also governed by ion channels. And the third, 2.5 is there are special proteins which are very important in metazoans, that is in animals like ourselves, which are integrins and caterins. These are molecules that sense the environment, sense both in an intermediate way between chemical and mechanical. How do they work? I'll just make a quick sketch at the blackboard again. So, as you might know, our tissues, epithelial tissues, that are the linings that cover all our body from the outside and from the inside, are organized in layers. And these layers sit onto connective tissue, which is the basis of our epithelial tissues. And so they are sitting on a layer, which is called the basal lamina. And this is a cell of epithelial cell which sits there. Now the question is, how does the cell know that she's sitting here and it's not some other place around? Because this is the correct location for an epithelial cell, and this is not the right location, either this one. Basically, when you're shed out, it means that you're losing contact, and then your skin just would fall into pieces. And when an epithelial cell starts crawling inside the connective tissue, well, that's something that happens when you have a cancer. So these cells must provide lining, so must be organized in layers, schematizing, of course, but they must know what's the inside, what's the outside, and they know it because they make contact with this basal lamina. But how do they know? Well, they have receptors for this. And these receptors are called integrins. And these work, you might think of them just like little hooks. So we make an enlargement of this picture. And so this is the basal membrane, and this is the cell. They extend outside the cell. And what do they bind? What's the ligand for these sensing molecules? Well, it's all sort of stuff that makes the basal protein. Fibronectin, collagen, all sorts of fibers that are present here. So these are receptors that bind chemicals that actually make a substrate, special chemicals that make a substrate. And in doing so, they also sense force. Because if you compress or expand this substrate, they will react. So in this sense, these are mixed chemical and mechanosensing receptors. And caterins, what are caterins? Caterins are the equivalent of integrins, very loosely speaking, which mediate the contact between cells rather than from cells, between cells and substrates. So cells know the presence of other cells at their sites because they have these receptors which actually connect cells and sense themselves forces between cells. So if you stretch your layer on one side, they will feel it and they'll try to respond by an opposing force, which gives the compliance and the elasticity to our tissues. So you see, things are pretty much mixing up at the level of molecular sensing. And finally, there's another very large class of receptors, which are tyrosine kinase receptors. The biological chemical mechanism that transduces signals in tyrosine kinase receptors is different from these other ones. It works according to other pathways, and they are very important in human cells, this transduction. Actually, they mediate most of the signals that tell our cells to stay in place, stay put if there's nothing to do, or if there's a wound, replicate, invade, and heal the wound, for instance. And defects in the sensing of these tyrosine kinase receptors are extremely severe, and the most known example where there are problems in signaling, mutations that impair signaling in these receptors, is cancer. So basically, the vast majority of cancers are related to mutations that impair the activity of tyrosine kinase receptors or of the pathways that descend from this signaling. So what comes after the sensing. So as you see, if you take very different standpoints, we can define different senses based on the organ, based on the stimulus, based on the molecular understanding of the receptors that mediate the stimuli, and they often mix up. So gustation is certainly in chemosensing, but it's sometimes mediated by some receptors, sometimes from others. This was just to make the point that looking at things from different scales might lead to different conclusions about what senses are. But nevertheless, we all agree that senses are well defined from the single cell level to the organism level. They are pervasive. So, yeah. Immune system definitely is a sensing system. You have to sense the presence of some outer organism and react to that. Yes. Yes, I'm trying to recollect whether the immune response is mediated. I think it's mediated by receptors of this class, but I might be wrong. I'm just trying to recollect. Okay, so sensing serves two purposes. Actually, it's one purpose, that is to connect ourselves with the environment. But of course, the environment contains also stimuli emitted from other individuals. So sensing is also important in communication. It's the receiver end of the communication system. So it's also interesting to compare different senses according to their typical properties. Now, here I'm using the classification in terms of signals. So you can compare chemosensing with acoustic sensing, with visual sensing and tactile sensing. All of these senses have specific features. Here I highlighted the sort of problems, the most problematic aspect of each communication system related to sensing. So tactile sensing is very good. It has the difficulty that you must be in contact with what you sense. So you cannot make distant sensing through touch. The visual sensing is, of course, is extremely good. It's highly selective and works on a rather good range, medium, depending on the organism, but typically it's medium. One problem is that it doesn't flow around barriers. If there's an obstacle, you don't see what is beside the obstacle. If you are in a forest, you won't see that there is a source 50 meters away from you. If you are in an open field, you might see it. But that makes the difference between life and death. Acoustical signals are very good. They can go a long way. They can flow around obstacles. But they're actually very expensive. For ourselves, maybe it's not that expensive. But if you go to the scale of an insect, think about a cricket signaling through acoustic signals for mating. Oh, that's enormously expensive. Sure. So acoustical systems are long range. Yeah. So the problem actually that this incorporates the fact that if you have a sparse obstacle along the way, you won't see. So visually in an environment, if it's completely free, okay, then vision is long range definitely, if there's no obstacle. But in a typical environment, well, you can see, I don't know, 100 meters away from you, okay? And then acoustic signals can travel longer, and olfactory signals can even travel even longer, up to 1 kilometer away from the source. Okay, you should take it with a grain of salt, but still, the point is that the great difference is that this is very short, and these are rather long-range communication. So this was just to tell that actually we as organisms typically need more senses in order to integrate over all these possible aspects and to have a more complete understanding of what our environment is. And the same holds for cells. Okay, cells need lots of information to come together, but actually cells are typically specialized at single cell level in one specific way of communication, which is the chemical one. This is really it. Sure. Could you please speak up because I'm... Thank you.
Sorry, you were mentioning about the energy costs for crickets being higher. Could you elaborate that a bit more?
Yeah. So crickets, as you might know, just emit signals to make mate recognition by actually the actual mechanism I don't really know it, but they move their legs in such a way that they produce sound, and this is metabolically. I mean, it's a large expenditure, an energy expenditure to produce this sound, as compared, for instance, other insects which communicate through odors. We will touch upon this at the end of the lectures, because they secrete very small amounts of volatile compounds that can signal over a very, very large range of scales. And so if you just compare the metabolic costs of sending an odor signal to sending an auditory signal, the second one is much more costly than the first one. Is that?
Yeah, but isn't the processing cost also different? Like for visual, I would assume
the processing cost of information?
Yeah, like for example, you'll have multiple layers and you have to do all sorts of contrast inhibition and all these things in visuals.
Well, about the sensory part, I will definitely talk much more on this. Yeah, the cost, I mean, here I'm talking really about the energetic cost at the level of ATP consumption or very basic information. So the cost of sensing is something which is more difficult to assess and typically is much smaller than the cost that you have to send the signal. Okay? So, but there will be more on this later on. So, time to wrap up about this long introduction, but I felt it was interesting to have a broader view on what the senses are. We shall be focusing on chemosensing, because it's the sort of mechanism that goes throughout from archaea to humans. And chemosensing, in particular, depending on scale, might be olfaction at our scale, or at the scale of insects and moths, which are actually exquisite examples of olfactory sensing. Then we might go down, and there is chemosensing at the level of single eukaryotic cells. I think Professor Cox lectured about this mechanism. So there's these lime molds which respond to chemical signals with the specific typical behavior, which is very important for their survival. And now we'll be also discussing a little bit about the mechanism of sensing in slime molds. And then we can go even shorter and go to the scale of a single bacterium or archaea, for what it's worth. It's basically the same thing. And how they sense chemicals in the environment and how they respond to them, which is what we call chemotaxis in these two cases. So during the lecture, I will spend most of the time on this problem, which is probably the best known at the molecular level. It's the simplest one also, but it's very, very rich in behavior. Then discuss a little bit what changes when shifting from prokaryotes to eukaryotes. And then, in the end, discuss about what happens when you just go to entirely different scale, which is our scale, basically, from centimeters up. So let's take a little guided tour through the history of this chemosensing issue in small organisms. So this story actually starts with bacteria, not because, of course, these are the first organisms where chemosensing has been discovered. I mean, we first discovered in ourselves that we can sense chemicals. But we start with bacteria because bacteria are simple enough and complex enough. Simple enough to have basically a very, very good genetic understanding of what's going on, what the hardware is. And you can experience with them very easily, which is also a good property. But their behavior is increasingly rich. Their variety of forms is extraordinary. You can have bacteria less than half a micron, bacteria that you can see by the eye. This is nearly millimeter. It's a single cell. It's hollow, but it's a single cell, which It's a single individual bacterium, nearly 1 millimeter. And then you can have all sorts of shapes and all sorts of external organelles. So they are both complex and simple at the same time. And then you can talk about something such as behavior in a bacterium. And so they are sort of very interesting from this viewpoint. And their story, actually, the story of observing bacteria is very, very old. And it started in the 17th century. So this is Antony van Leeuwenhoek, who actually was the first one to develop a microscope, a set of lenses in which you could see small things. And so he wrote these letters to the Royal Society describing what he saw. And he saw many incredible things. And actually, if you read extracts from these papers, it's also very funny because understandably, he had the conception that what he was seeing was just organisms such as ourselves or animals, but just writ small. So he's trying to see where the legs are, where the nose is, in this small objects that he saw. And from the description that he gives, and which are quite eloquent, we can actually understand that most of his observations were related to a bacterium which is called Spirillum volutans. And the typical thing that he observed is that these small animals, animalcules, he called them, actually can swim in water. And so this is a movie taken in a lab, and you can see these small bacteria, not so small, actually, they are 50 microns, sort of already on the large side for bacteria in length. And they swim. How do they swim? Well, we'll get on that later on. But definitely they do something. And van Leeuwenhoek already noticed that they sort of swim so on purpose. Because if you disturb them, they will change their motion. Van Leeuwenhoek was very imprecise on this point, but definitely said, okay, they can swim and they can do so in one way or another, depending on whether I put them in one condition or another, which is a sort of first intuition, insight that this behavior of swimming maybe can be modulated in response to some external stimuli. So the next big step actually had to await a long time before to occur, and we were already in the 19th century. And the first interesting observation is that in this particular bacterium, Chromatomocani, which again is pretty thick bacterium, which you can see at the microscope even with the abilities of the 19th century, early 19th century. It's red, bluish red. You can see here. It's red because it has these vesicles inside which contain sulfur. And it's sulfur that makes it red. Herrenberg himself was in the wrong conviction that all these were internal organs, so he was able to detect the lungs, the ovary. Okay, so well, that's understandable. But he made the first observation that actually these large bacteria, which also swim, they do so because they have some sort of appendage which propels them. So that's the first observation, qualitative, of the presence of flagella. There will be more on that, but flagella are some external organelles that give the bacterium the thrust to swim through water. So that's sort of putting some mechanics into the motion of these organisms. And then came, at the end of the 19th century, the first clear-cut demonstration that these swimming bacteria respond to stimuli. And that's actually a very impressive piece of scientific work. This scientist, Theodor Engelmann, was one of, in the 19th century, you could be a physiologist and a musician at the same time. He was a member of the Wagner group and was a very good musician. And he also made incredible contributions to physiology. Actually, he was the first one to understand that chloroplasts in algae and in plants are the places where photosynthesis occurs and oxygen is released. And how did he do that? Well, the first he discovered that there are some bacteria which respond to specific stimuli. So his first observation was that what he called the bacterium photometricum, so the bacterium which can measure light, and probably you understand that it's this Allochromatium venosum bacterium, is afraid of the dark. So he was looking at this bacterium swimming into his microscope. Then he moved the hand in order to just put some shade on the place where bacteria were, and bacteria were just moving away from the shade. They had what he called, they are afraid of the dark. A phobic response to the dark, he called them, and just interpreting psychologically what the bacteria did. But actually, what the bacteria, specifically the bacteria does, is that it's looking for light rather than escaping from the dark. And there are other bacteria which he observed, and these are the original drawings. So let's just take a couple of minutes to understand what this experiment is. So he took these algae, Spirogyra, which are characterized by the spiral arrangement of chloroplasts. And chloroplasts are the organs where photosynthesis, of course, we know it now. Then what he did, he put these algae in a little pond with all bacteria inside. And then everything is in the dark. And then he's illuminating two different spots along the algae. In these spots, there are no chloroplasts. There are no these green structures. He illuminates, and you see that bacteria don't go there. But then he illuminates the region where this chloroplast here, and bacteria tend to go there. And then if you illuminate everything, bacteria congregate along the spirals. So he argued, okay, my bacteria love oxygen, because if I provide them oxygen, they go where the oxygen is. And then they like to go there. Then this is the place where oxygen is produced in algae. And he was right. Might have been wrong, but it was right. Okay, so this is a first definite example of small bacteria swimming in one direction, in one specific place, because there's something that they like there. So shortly after these findings, people started to put up some more definite assays for this phenomenon. And this was done by William Pfeffer, which was known for other works in plant physiology. And he coined for first the term chemotaxis, that is, taxis, movement, Greek, induced by chemical substances. And he was the one who invented actually the capillary assay. What is the capillary assay? It's a very simple assay, basic to detect whether some microorganisms respond to certain chemical stimuli or not. And first up, detailed application of this chemosensing was actually at the very end of the 19th century, where using the same chromatium Ocani that was discovered by Engelberg a few years earlier, you see that there's a capillary, so it's a very small tube, which is filled with some chemical, and then you put it inside a pond, a small pond where there are bacteria, and you observe what bacteria do. And you see in figures A and B, which are original drawings by Miyoshi, bacteria go towards the mouth of this capillary and then start to move in bands up. In this case, it was ammonium nitrate. But if you change chemical and you put malic acid, they tend to go away. So bacteria sort of have a sense of taste. There are things that they like, things that they dislike, and they swim and get away accordingly to the stimuli. Okay, that's very, very basic but important information. And then, at the end of the 19th century, all this interest in motility and chemical responses of bacteria completely fades away. Why? Well, because it starts the golden age, so-called golden age of microbiology. These are the years where Koch, Pasteur start to work, and in a few years, they discover the presence of this very known pathogenic bacteria, their influence that they have, Pasteur starts doing immunization vaccines. So everything blooms in this sector, pathogenic microorganisms. And microbiology has been for, say, 50 years, and then just a problem of pathogenics, vaccines, and how did we deal with this organism. Very, very scarce interest in motility. Actually, in hindsight, now we know that there are pathogenic species which are motile and which are not. So motility is not quintessential to pathogenicity. For instance, Bacillus anthrax, which is an extremely aggressive bacterium, in the sense that you need very few copies of this bacterium in order to get infected. It's very virulent, technically speaking. Doesn't move at all. Actually, it's carried away by the wind. It floats. Other Vibrio cholera swims very actively in water. But so there's no specific correlation between the two aspects. And it's not that you're motile, you're pathogenic, or you're non-motile, you're not pathogenic. You can be pathogenic both being motile or non-motile. But if a species is pathogenic, being motile adds to its pathogenicity. So it's a second-order contribution to pathogenicity, which comes from the ability to invade the organism. So because of this reason, sort of the progress in chemosensing and chemotaxis sort of lagged behind for many, many years, with some few sparse exceptions. One of them, notable one, was the contribution given by Buddha, who actually was the first to make sufficiently detailed observations in dark field microscopy to see effectively how Chromatium ocani, the bacterium that we encountered already a couple of times, how does it swim in water. And how does it do? Here you can see this movie. You can probably see it. Well, that's the cell body, which is fluorescent. And here you see, you don't see. You will see again in a second. I hope so. I know it's blank. Well, yeah, it is. So in the back of the bacterium, so to speak. Any clue about that? No. In the back of the bacterium, there is this organelle, which is sort of a helical structure, which continuously rotates and propels the bacterium like an helix, like that. Actually, it's not a single flagellum. You wouldn't be able to see with this microscopy a single flagellum. What is it? Actually, it's a bundle of about 40 of these flagella, which are all sitting in the back of the cell. So you have this cell, which is, as I said, a large one. And then at one side of the cell, there are all these flagella that make a bundle. There's 40 of them. So it's pretty thick, actually, visually, because there's many of them. One single flagellum is just a few nanometers wide. But when they come together, they are thick enough that you can see it at ordinary microscope. Sure. Yeah, so yeah, that's a long story. We'll be discussing about this. Actually, the point is, one important point is that they are localized at the same point. So basically, when they come together, hydrodynamic forces are keeping them tight. So they rotate all in one direction or all in the other direction, because that's hydrodynamic forces. Sorry? It is triggered from internally because there's a signal which tells all the motors to rotate in one direction or the other. Okay, so that's the signal that comes from inside. Oh, there are motors here. We'll be elaborating on this soon. There are motors that make turn this flagella from the inside. And these motors respond to internal signals. And these internal signals change in time, actually, typically in default. So it may be turning one direction or the other. So the bacterium might be going in this direction and then opposite one, always with the flagella in the back. But what keeps this thing together outside is hydrodynamic forces. So there are two things. The signal must be synchronized from the inside, but also outside the forces might not be disrupting this thing. So, but there will be more on flagella later on. And so at that time, they started also to understand what are the physical mechanisms of propulsion, how the forces may be exerted. But since I will expand on this in the specific case of E. coli in the following, I will defer this discussion of propelling forces to the following. So after this long period of inactivity about chemosensing and chemotaxis, then the new era, the modern era of chemotaxis starts in the 60s, 1960s. Basically due to a series of work by Julius Adler. Actually, by rethinking all the assays about chemosensing and chemotaxis, he was able to discover a wealth of phenomena, including probably the most important, the fact that the bacteria, and in particular E. coli, have chemoreceptors. Okay. So this goes on with two basic biological tools, two assays. The first one is the chemotactic swarm plate assays, okay, which we will see in a second what it is. But the basic idea is that you put your bacteria into a petri dish, which is filled with some agar, but it's still liquid, so bacteria can swim in it. It's not so solid. Then you deposit some bacteria in the center of the petri dish, and then you just see what happens. The petri dish is filled with nutrients, so you expect this bacteria to grow, and then you make quantitative observation on how this thing grows. And then you start doing the usual game of genetics. That is, you start producing mutants, and you look for phenotypes which are different from the usual ones. And these phenotypes are just the way these colonies develop. You look them by the eye and you see, okay, this is doing something strange, then it's a mutant, and I will pick it up, and so on and so forth. So by this way, you discover mutants which are related, and genes which are related to the chemotaxis and chemosensing. So what is the swarm assay? So let's take some time to look in detail at these figures. So this is one petri dish, and there are actually many colonies in the same time in the same figure, so we'll take some time to go through all of them. So that's the basic, the sort of default or control case. You have put your bacteria in an inoculum here in this point, and then you wait for hours, typically. These fronts that you can see propagate outwards as circles with a speed of roughly micron per second. Okay, so it takes a few hours to make out centimeters. And then you start seeing different structures. So what are they? How is this? Why are these concentric rings appearing? So the basic idea is that as this bunch of bacteria progresses outwards, it starts consuming food. In this case, the bacteria are put into a medium which is actually a mix of several different amino acids. So what they do, they start moving outwards because they swim. They eat nutrient. They actually consume it all, and they leave no nutrient beside, at least. They start with serine, an amino acid. So outside this ring, there's plenty of serine. Inside this ring, there's no serine at all. Everything has been consumed. So as they progress, they eat, and they leave nothing behind. They are really just invading pests that eat out all the crop as they go. Second ring, okay, they're short of serine. Okay, serine is my favorite amino acid. Too bad, it's over. Let's switch to something else. It's going to be aspartate. And then a second wave starts, and all the aspartate is eaten out. Next, threonine, another amino acid. Next, tryptophan. And he made a list. So E. coli has a perfect list of wishes. If I'm faced with a menu at the restaurant, I will go first for chicken, then for, right? So how can you check that this is exponential? Of course, you look for mutants. And you see, if there's a mutant which is lacking for some components of the machinery, we don't know that yet, what it is, that consumes aspartate, you will only see the serine ring and the more diffuse threonine patch inside. And if it's missing serine, you will see only the inner ring. Notice that this is not exactly the same as this ring, which means that there's crosstalk between the different nutrients and different responses. And then if you have some mutant which is not able to swim at all, that's what you see. So you see, in order to produce these patterns, there must be swimming, that is spontaneous motility, and sensing. The ability at least to sense, to respond, and here also to consume, because you just consume. On this other plate, it's pretty much the same experiment, but now this is filled with solution where the nutrients are sugars, ribose and galactose. These two inoculi, and you see that these two rings propagate. So in this lower ring, it's a mutant that doesn't eat ribose. In the upper ring, there's a mutant that doesn't eat galactose. So they just don't talk to each other. And as the ring progresses, they just go through each other without interacting, because every one of them sees a different environment and doesn't care about the other sugar. Okay? Very simple experiments, but with an intensive genetic work of producing mutants, there's unveiled lots of very interesting things. And lots of questions also at the time, because you would like to know what is important in chemotacting, so in moving outside. Is it important to consume, to sense, only to sense and not to consume, only to consume and not to sense? There are two mechanisms here. You consume and you sense. Which is the relative weight of these two? How can one discern? So what Adler did is that he developed a more elaborated version of the capillary assay, but the spirit is the same. You put into a capillary a nutrient. Here you have bacteria, and then you just count how many bacteria climb up and how they do up this capillary. And then if you look at the number of bacteria that you can capture into the capillary as a function of concentration of galactose, you can see the response as a function of concentration. And you see that the response is important in a wide range of concentrations. Here is this galactose plus. We'll get to the mutant in a second. And you see that there is a very important response from the micromolar to the millimolar range, which is broad range of concentration, where with a peak in the middle, right? These are physiological concentration of galactose that typically induce the best response. Now, what Adler did is that he produced mutants which are defective for uptake and for the metabolism or galactose. So these new mutant bacteria were not able to import galactose and were not able to metabolize it. So this sort of, if you put them in a beaker and then you measure galactose as time goes by, it stays always the same. They're not able to change their environment. And so you ask, what happens if I put these bacteria, which are not able to uptake and consume, into my assay? And what he observes that they swim up. They do. They respond in a different range of concentrations, but they do. Which means that they have a sense of smell. They go up because they feel the presence of this good thing, even if it's nothing to them from the metabolic point of view. They cannot make energy out of it, but still they go. And this is the first proof concept that bacteria have chemoreceptors. So they must have some chemical structure that senses the presence of galactose in this case and enables them to swim up. Then you might ask, okay, why is this sensitive to this concentration and when it consumes to this one? Well, that actually makes perfect sense after a moment of reflection, because when you consume, actually, you just sort of dig a hole in the concentration field, and then it's different, actually, effective range of concentration that you are experiencing. In order to make it clear. So if you progress in a profile of concentration like this, so this is galactose, plus galactose minus galactose. If you are the mutant, you don't alter this profile as you swim. So there is this wave of bacteria that goes up, it goes up, it goes up, but the profile stays the same. Okay? And so you respond to a rather shallow gradient. Now, if you consume, what happens? You just deform your place. There is a saturation at a sensitive level, but there's also the fact that when bacteria are very, very well fed, they start swimming, right? So they don't even, if you look at them here, they don't swim. So they cannot, no matter what you put, they can just swim up. There are two effects together. Okay, so by doing this extensive work, actually Adler was able to come up with a very detailed list of all the chemicals that the bacterium can sense. So it can sense basically all amino acids. Amino acids are very good nutrients, as sugars are, and alcohols as well. But it can respond also to other things. Okay, the peptides are basically couples of amino acids, but also other things that you might not have guessed from at first sight. And also, there are several responses which are repellent responses. Chemicals that bacterium doesn't like, and so tries to flee away from them. And there are also some amino acids which are bad to bacterium, and there are also more complicated situations where some chemicals are attractive at some concentration and repellent at others. So depending on the concentration, you might wish to go there or flee away. So from these studies, actually, there's been, again, a new flourishing of studies in chemotaxis. And just to give you an idea of the many things that one can observe in a simple experiment, such as this warm plate assay, here I'm showing the sort of patterns that you can observe in such systems just by changing a little the conditions of the medium. So it's basically the same experiment as before. You just seed in a certain number of bacteria in the center of your dish, and you let them swim. Only in this case, what happens is that the medium is filled with fumarate, which is not a chemoattractant. So bacteria shouldn't form a ring. They should just stay put, or expanding, but very slowly, without the aid of chemotaxis. But the point is that when bacteria eat fumarate, actually, in the metabolization process of fumarate, they produce aspartate, which in turn is a chemoattractant. So these bacteria start to respond to the signals sent by other bacteria as they consume. And what happens is that each bacterium is producing aspartate. So other bacteria are seeing that and trying to go where the aspartate is. And so they tend to cluster. This homogeneous distribution of bacteria is unstable dynamically. And no bacteria tend to collect them in these small dots that you can see here. So as the wave propagates out, they eat fumarate, they produce aspartate, and they aggregate. Very short old movie that shows this 30 years ago. You can see it's already started. The ring propagates out, and then in the azimuthal direction, you can see the formation of these structures where bacteria are coalescing together because of their relative attraction due to aspartate production, and so on and so forth. And then depending on various parameters, you can find different arrangements of the spots. And then if you go to different setups, different substrates, different bacteria, you can find all sorts of patterns, very complicated ones. But actually, all this richness of patterns is basically given by the fact that bacteria consume and modify their environment and respond to it. That's the basic mechanism. And here you can even have these swirls where bacteria are turning around in a given direction, sort of bacterial vortices on the swarm plates, and all sort of interesting phenomenological aspects. So, as you've seen, all these experiments are basically looking at bacteria as populations. So we're not looking them at individual behavior in the swarm assay, in the chemotaxis assay, in the capillary assay. They're looking at them as distribution of bacteria in space. So one of the first concerns of theorists, of physicists, is how can we model the evolution of such populations mathematically at the large scale level? Okay, so this is centimeters. So the very basic idea is that one can cook up phenomenological models that are able to describe the evolution of these profiles, density profiles of bacteria. And the basic mechanism actually was put forth initially in the 50s and then revived by the Adler experiments and, in the 70s. And it's a very elementary, actually, mathematical model, which considers two spatial distributions, which is the number density of bacteria, U. This is just a measure of how many bacteria you have in 1 millimeter cube number, which depends on space. And here is the distribution of resources. Concentration of galactose at a certain point in the swarm plate, in the Petri dish. And then you write down equations for the evolution of these two quantities. So let's try to see qualitatively what these terms mean. So let's start from the end. So this is a term which says that there is a change in number density in a given position of space. Okay, all these are differential operators, gradients. That is, they take differences in space. So this term does not take any difference, so it's something that depends on the place where you are. And what is it? Well, this is just the growth rate of bacteria in a given place, which depends on how many bacteria you have, and it depends on how many nutrients you have. So this function is, in general, maybe quite complicated, but in some instances simple. For instance, if you are in the exponential phase, it must depend linearly on the number of bacteria. And if you are in the nutrient-limited regime, diffusion limited, it should depend linearly also on the concentration, on local concentration. When there are lots of bacteria, then this turns to more complicated behavior. When there's lots of food, also it may change. So in general, it's a complicated function, which basically tells you how many bacteria will divide in the unit of time. And then these are the terms which are related to space, changes in space. This is a term which describes diffusion. That is, random, unorganized motion of bacteria. If you put them to swim without any particular directional cues, so suppose you erase all possible chemotaxis genes or sensing genes, they just consume and swim, then this term won't be there, but there will still be this term, which means that bacteria swim, but in an uncoordinated fashion. And then you model this as diffusion, ordinary diffusion, but of course with a coefficient which is not the thermal diffusion coefficient of bacteria, which is very small, but it's sort of effective diffusion coefficient that will describe these agitation internally due to bacterial motion. And then this is purely the chemotactic part, which tells that besides this diffusive unoriented motion, there is a directed motion in the direction of the gradient of concentration. So if there's locally around them, the bacteria see that the attractant is increasing in that direction, on average they will go that way. Look, at this level, this is a completely phenomenological description. Sure. V is the nutrient distribution of resources, nutrients, concentration of glucose, for instance. There's no fluid, and there won't be at any time. No, sorry. There's no description of fluid motion here. Absolutely, it's totally uncoupled. The fluid is assumed to be at rest. Of course, it's not at rest because if you swim, the fluid will move. But you're totally neglecting this aspect. If you put it into, many interesting things appear, but it's the story of the last few years. Thank you for the comment. So the interesting thing is that if you study these equations from a mathematical viewpoint, you find that they admit what they are called traveling wave solutions. So if you start with the localized distribution of bacteria, it will move out. In this case, it's just one-dimensional, just like a capillary assay situation. So you start with the distribution of bacteria. Actually, this has already evolved, so it was distribution of bacteria here. And then it moves to the left. It eats out the nutrient. So this front of nutrient availability regresses in that direction, and then the bacterial front progresses. And then you can show that these equations, for a certain class of functions that you choose, not all of them, admits traveling wave solutions with fixed speed. So this is a sort of mathematical demonstration that these models can capture the actual behavior in a capillary assay with very few ingredients. And of course, you can go to the computer and try to see what happens if I cancel one term at a time, and you see that they give different behaviors, different shapes, different results, and then you can go back to the experiment and check, and so that's, sure. The second equation? Right, that's it, exactly. Yeah, the gradient is like this, so particle will try to go like this. Right, so it tends to broaden this distribution. The last term, it's a growth term, of course. Then it will let bacteria grow very little here. So bacteria won't grow here because there won't be any resource, right? And they will grow like crazy like here because there's plenty of resource. Okay, so this also produces a push in this direction, right? So it's interesting to see what happens if you just drop this term. If you drop this term, and then you make reasonable assumption about this. This goes under the class of what are called the Fisher-Kolmogorov problems, systems. Then you will have the same thing like this, except that this whole thing will be broadened because diffusion, and then it will just fade out very, very slowly. In some cases, it will even fail to give a self-propagating solution just because of this trailing edge. But then chemotaxis just helps bacteria to keep up and then put them together in a short ring, in a thin ring. Satisfied with that? Not really? Moment. Chi is the chemotaxis function. Okay, in the next slide I will show you examples, but first order approximation is it's constant. So it starts with a constant for low densities and low concentrations, and then it might be modified. Again, this accounts, for instance, for the fact that if you have very, very much nutrients, you don't respond. Okay, the fact that you don't move at all is included in the dependence of this on concentration. You can put all phenomenological effects, but I want to stress that this is strictly phenomenological. So in fact, when you put these things to the test, you're actually able to reproduce the patterns that you see in the fumarate aspartate system at some price. Because as you see, the functions that appear here, so that's the chi function that you are evoking. You see, it's a function. It's not easy to get this from molecular biology. But as I will show in the end, we can get to something like this, starting from molecular information. Not exactly this one, but there's a possibility of bridging the molecular small-scale information to the macroscopic information, description, sorry. So you see, you plug in specific forms. Okay, these are not just taken off the top of my head. People can do experiments when you try to sample the rates of growth as a function of, you do what are called chemostat experiments, when you put bacteria and then you see the rate at which they grow if you put tot molar of this thing, tot molar of this other thing, and then these things you can actually measure them. So it's not completely nonsensical. You can measure them, you can measure parameters, not all of them. And you see there are 9 here. Sort of tricky. Some of them you measure, some of them you know, some others you guess. But that's a phenomenological model. At some time you should put something like this. But nevertheless, you can test, sure. In the chemotaxis function, actually, there's only dependence on C, because these are mixed up, sorry. So n must be there just because it's the bacterial density. So the coefficient is just K1 over K2 plus Kc squared. Yeah, sorry, that was confusing, my fault. Where n appears explicitly is in this growth rate. So this is the carrying capacity, what is called. So the maximum number of bacteria that you can host in a millimeter cube given a certain concentration. And then here is the consumption rate. So the rate, sorry, here is the consumption rate. And this is the production of aspartate via fumarate. So you should put all the ingredients inside. Okay, very good. So that's sort of, we've been doing this thing chronologically in order to see how people have come to that. So first the observation at the microscopic level, then the observation of behavior at the macroscopic level, and then reverting back now to the microscopic level, trying to understand how does this process works at the level of a single cell. So let's take again our magnifying glass and get back to the bacteria and look at them in detail. And the bacterium that everybody uses when just wants to play around with this chemotaxis phenomenon is E. coli. Chemotaxis is not at all restricted to E. coli. Basically, all bacteria have the same machinery for chemotaxis. We restrict to this because it's the most studied example, the simplest one, but you shouldn't be thinking that all that I'm talking about is specific to E. coli. It's generic to bacteria and archaea. But now follow some specific facts about E. coli. E. coli is an organism that typically inhabits our guts. It's not the most prominent one, actually. It's just 1% of our microbiota made of E. coli. It has been isolated in the beginning of the 20th century, from babies, actually. This strain, K12, is a friendly strain. It's a strain that lives in our guts and eats together with us. And our microbiota, so the ensemble of all microbes that live in our guts, actually are typically very helpful to us. And they help us manage with many, many challenges that are put to our digestive system by the foods we eat. This strain is so friendly, actually, that since it has been first isolated in the lab and cultivated there, it has totally lost the ability to colonize our guts. So even if it's good, it cannot re-enter in our guts, which makes it also very, very good for experimentalists. But there are variants which actually can be very, very nasty. This strain is potentially lethal. It can produce hemorrhages of the urinary system and lead to death in a couple of days. Okay? So it can be either nice or bad to us, depending on the strain. It's a relatively small bacterium. Its size is about 1 micron cube. When it's well fed, and you provide it with amino acids and sugars, it doubles itself in 20 minutes, which is sort of maximum replication speed that you can have for an organism of that size. So which means that you can produce several generations, you can produce several mutations, and then it's an ideal playground for cell biologists and molecular biologists. The colonies can be as dense as 10 to the 9 cells in a millimeter cube, okay, which makes for a population in a centimeter cube, so a population of India in a spoonful, basically, by numbers. And its genome is being sequenced and resequenced many times, and we know all the details about it, the sequence, except that for many genes we don't have a slight idea what the function is, already in E. coli. Maybe they work in some conditions that are not present anymore. We don't know. Current studies still ongoing on this. So that's about the sort of ID picture of our bacterium. Now, for us, what is of interest is that this bacterium, as all bacteria, is an envelope, a cellular envelope. It's actually a 2-layer envelope for E. coli. What is inside the cell is a sort of relatively unstructured mix of cytoplasm and DNA, and of course all the machinery that serves for transcription and transduction. But there's no very, very clear-cut compartmentalization inside a bacterial cell. It's sort of a soup, dense one soup. But it has many interesting external organelles. So structures that are outside the cell body. And one of them is the flagellum, which we will be mostly interested in. So the external organelles actually serve the bacterium with the purpose of making contact with the external world. And there are 3 big classes of external organelles. One of them is the fimbriae or pili, which are these appendages. These are expressed only at special stages in the bacterium life. And they actually serve to make contact with rigid surfaces, contact bacteria. Bacteria can have an interesting social life. They can aggregate together, form biofilms, colonies, then spread again. And all these organs help to sense the presence of a surface. So, but there's also mechanosensing at the level of bacteria, which we won't discuss at all. But it's something which they have a sense of touch, and a selective one also, favoring rigid substrate with the presence of sugars rather than others, for instance. But that's totally entirely different story, so we won't be discussing it. Bacteria, which typically reproduce clonally by division, from time to time might decide to switch to sexual reproduction. The advantage being that it's their only way to make recombination. So to cross genomes, introduce variation, explore different landscapes from the viewpoint of evolution. It is not frequent, but from time to time bacteria can enter in this state of competence where they can exchange DNA material. And then our star, the flagellum, which is this external organelle, which has a spiral-like shape, helical one. It's not a whip. At variance with flagella in other organisms, which are flexible structures, these objects are pretty rigid. They're not totally rigid, so depending on the flow, they might switch from one certain type of coiling to another, but typically they're more akin to a corkscrew rather than a whip. Okay? And these flagella are the things that provide motion to bacteria. So let's have a look. Let's have a look at bacteria swimming in this very, very old film. So at the initially we will see them swimming close to a glass surface. So they are put into, under the microscope into a small petri dish. And then you will look in them which are swimming close to the surface of the bottom of the petri dish. And then focus will be on those that are inside the bulk of the motion. Oh, sorry. Okay, so that's what typically you see. You see some of them attached to the bottom, those that don't move, and the other ones swarming around, moving. And if you look carefully, they mostly turn to the right. And there's no specific rule of vehicle conduct at that level. So they turn to the right because of the hydrodynamic interaction with the boundaries. Since these flagella are turning into one direction, this sort of breaks the symmetry of motion and leads them to turn typically into this direction. Okay? But then when you go to the bulk, which is the second part of the movie, the motion is less organized. Let's play this again. Now we're going to bulk, and you see them changing direction, etc. But they definitely swim very fast. So how do they manage to swim in practice? Well, that's actually sort of a riddle. Because if you go to study the fluid dynamics at the scale of a bacterium, it's very different from the one that we experience. So you should take a completely different take at the problem from the one inspired by your own experience when you swim, for instance. So the motion of a fluid at our scale and at the scale of bacterium is governed by the same equation, which is the Navier-Stokes equation, which is nothing but Newtonian physics applied to fluids. Fluids that are not compressible sound waves, we can neglect them at our scales of interest. So that's the description that you have. And an important parameter that describes fluid mechanics is the ratio of forces due to the viscosity, that is the resistance that the fluid opposes to you moving into it, to inertia. And inertia is your ability to coast. You just start and then you let yourself go. So this ratio of these two forces, resistance to coasting, is encoded into a single number, which is the Reynolds number. It's an actual number, which for bacteria it's of the order of 10 to the minus 5, if you put the figures in water, which is extremely small, which means that bacteria experience a very viscous environment around them. When you swim, your Reynolds number is 10 to the 5, plus 5, 10 orders of magnitude larger. So it's entirely different. In fact, in water, you can coast. You just take a stroke, and then you let yourself go, and then you go 1 meter, 2 meters, depending on your power, just without exerting further force. It's inertia that brings you onward. But bacteria don't experience this. If the flagellum were to stop at a given time, the bacterium would stop immediately, basically. Just a few nanometers and then completely stop, because the resistance is very, very high. It's just like we were swimming in molasses. Think about that. But it's even more viscous if you put the figures. Okay, so that's a completely different environment. And it gives rise to sort of not very intuitive behavior. So this is a classic experiment in the fluid dynamics of small Reynolds numbers. In this beaker, there is a highly viscous fluid, say silicone oil. And now the experimenter, which by the way is a renowned fluid mechanicist, which is G.I. Taylor, just puts a drop of ink inside. No, actually it's oil, it's the same oil, but colored with ink. It leaves it here, and then starts stirring the fluid, and as you expect, the ink mixes, and after a few turns, you can only see this blurred image of the remaining ink. You make a certain number of turns, then you start turning into the opposite direction. And as you turn, and you turn, and you turn, what will you see in the end? The droplet gets back to its place. Okay, now you can try this at home with your milk and coffee. Will it work? No. That's the Reynolds number, entirely different. Now you can have a grasp on how this happens just by looking at the equation, in fact. Because if viscosity wins over velocity, over inertia, your equation drops completely this term, and then you're left with what's called the Stokes equation. And this Stokes equation is completely invariant under time and velocity reversal. Turn the arrow of time, turn the direction of velocity, you get exactly the same solution. So, low Reynolds number fluid dynamics is the word of reversibility. It's a word where Aristotle is right, because force is proportional to velocity and not to acceleration. And it's a word where going forward and backward in time is the same thing, which implies that if you try to swim into a high viscous fluid doing like this, like something like you do you can do in the pool, you do like this, then you this, then you like this, then you this, won't bring you nowhere because you can't coast. This is what is called the scallop theorem. So you need an entirely different modality of swimming in a microscopic environment. Okay, so we'll, sure.
Why doesn't the bacteria go in a streamline fashion when the Reynolds number is so less? Why does it execute a random motion?
Sorry, can you speak into the microphone?
Why does the bacteria go in a streamline? I mean, why doesn't the bacteria go in a streamline motion when the Reynolds number is so less? Why does it execute a random motion?
This
Like,
bacterium does a random motion when you see it from a large scale. When we will see it on the scale of a micron, it will go straight. And then from time to time change direction, if that is the question. So we see random motion just because we're looking at a distance. Okay? But if you look at it locally, it will go like this, and then turn, and then go like this. So there will be streamlined motions interspersed by changes of direction. But there are other bacteria which perform continuously streamlined motion, just like steering like this. There are others which go straight, then stop, then go back. So there are different modalities of motion. But the, the description in terms of diffusion is something like a large-scale description, when you coarse grain over all these details of this going back and forth. But there will be more on that on the next lecture.
So in this experiment, you have shown that an oil droplet, when you mix it in counterclockwise and clockwise, it comes to the same position. So can I expect the same thing with the microbe when I view it in the micro scale?
Yeah, definitely the same thing. Yeah, in the micro scale, I mean the Reynolds number is a combination of different things, right? So here in this experiment, we have human scales for velocity and length scale, but you have a large viscosity. Bacteria have a small viscosity because they are in water, but they are small and they don't go very fast. So that what makes small their number. So it's a combination of three factors. Viscosity is important, of course, as a physical parameter, but of course, if you go to small scales, then you will be much more likely be in the situation of low Reynolds numbers. Okay, so I think it's time is run over, and we will keep on talking about this next lecture. Thank you.